All right. So let's talk about the implications of chapters 10 through 12 and some PChem papers that relate to those topics. And also there's one at the very end that relates to everything. It actually reaches all the way back to, I think, chapter 1 or 2. But it talks about how kinetics relate to that. And of course, we had to wait until we were done with chapter 7 to talk about kinetics. And so the first thing I want to talk about is, you know, I don't want to go so far as to say that all biology is reducible to chemistry. But there's a lot of chemistry that's in biology. And a lot of times, even the biologists don't really know about that. So there's actually a chemistry of organs that R.J.P. Williams talks about. And I want to bring that out a little bit more to talk about the biology and the chemistry and how they go together. So we had the two great oxidation events and the atmosphere filled with oxygen. Then oxygen was available in large amounts to use for energy and for structures. And that's usually where I leave it. You know, the book allows me to go into a lot more detail about what those structures were, you know, what the energy was. But the structures actually allow different biological structures. Because just like the inside of a cell was specialized to have different compartments that do different things, inside animal bodies, and to a certain extent even plant bodies, but mostly animal bodies, you had groups of cells that got together and they had a shared function. Because they had a shared function and a shared developmental origin, they also have a shared chemistry. And that's the thing that is something that I want to think about. How is chemistry inherent to particular organs? Because certain organs have certain jobs. The big thing about structure is that oxygen allows cross-links. You know, it's got two bonds coming out of it. And so it's able to basically make lignin if you link sugars together with oxygen. And it's able to make connections. And so it's really good at making connections between cells. And that allows organs to form. That's the one thing that you really need. You need the cells to stick together if you want to have an organ that does a particular job in a particular space. Even beyond oxygen, you can see zinc and copper have particular roles here. Zinc allows cells to cut the connections loose when necessary so that cells can move over and start something new. This is good in the case of organ development, but it's bad in the case of cancer. So you see zinc doing this job outside the cells and doing a catalytic job that we see that is cutting rather than forming bonds. Copper, on the other hand, allows brand new kinds of oxygen chemistry at this point, and you'll see a lot of this chemistry reflected in different organs. So you have different compartments that are large and small that can hold different chemicals. Before organs developed, you know, you had cells which are a certain size, and you see right here, a cell can be up to 1 centimeter. I think technically like an egg is a cell, you know, and things like that. But most of the cells are like a period or smaller is how big they are. But organs are definitely up to 1 meter big. Whole organisms are up to 20 meters big, I guess if you have a blue whale or something like that. And then vesicles are a smaller size, and they're smaller inside the cell. Vesicles can be up to about 1 millimeter big, but they always have to be smaller than the cell that they're in. And so you have these 4 different sizes that have 4 different metric scales. And that's sort of a PChem way of looking at things. Okay? So the important thing to talk about, when we're talking about organs, we're talking about a compartment that does chemistry. Well, inside cells, you have organelles that do particular chemistry. And just remember that mitochondria are inside cells, and they look like they even have their own DNA. They look like they have their own chemical function. You know, and they look like they have their own chemistry as well. What's really interesting is the mitochondria is the place where you do oxygen chemistry. You don't do oxygen chemistry in the cytoplasm. The same thing is true for photosynthesis. It's the opposite oxygen chemistry, but it's still oxygen chemistry, and it's done in a chloroplast. You can do oxygen chemistry outside a cell. The one place where you don't really do oxygen chemistry is the cytoplasm itself. You have the idea that maybe the mitochondria a long time ago was a bacterium that could use oxygen, that had that job, and then it was sort of endosymbiosed, which means it was just engulfed by the surrounding cell. And then you had, the cell was able to set up, instead of breaking down the mitochondrion for food, it was able to set up sort of a mutual assistance program where they were able to send oxygen to the mitochondria and to take energy from the mitochondria. Similar thing could have happened with chloroplasts. The interesting thing also is how often were these endosymbiotic events, and that's actually a major debate. It looks like chloroplasts happened at least a handful of times. Mitochondria might have only happened once. And just like with the origin of life, how common are these events? The answer is they're probably not very common, but when they happen, they're really good. And so that's just an interesting debate. If you want to get into that in your final talks, maybe you'll be able to, but more likely you'll just file it away as a mystery right now. But realize that these subcellular structures can be looked at like organs. If you think of it as mitochondria are doing oxygen chemistry, they make ATP and they turn acetyl-CoA into O2, this is the subject of Biochem 2, into water and CO2. The chloroplasts do the exact reverse chemistry for photosynthesizing plants, and they have the light harvesting complexes. They have a lot of other things that support this chemistry. But realize that they are kind of reverses of each other, the red mitochondria and the green chloroplasts. And what these have in common, they have DNA. The DNA is a circle like it is in bacteria. They have their own membrane. The lipids look bacterial to us. And they even reproduce like bacteria do when the cell divides. They bud off from each other and they reproduce asexually. So we put this together with the fact that some bacteria can eat each other, they can engulf each other, and it makes sense that this could have been an ancient bacterium that just stuck around and continually reproduced itself inside the other cells. And over time, they got to be dependent on each other so that you can't separate the two anymore. One key piece of evidence for all this is the fact that the cytoplasm is still a reducing environment. Remember that these oxygen chemistry things are being done either outside the cell or inside an organelle. In the cytoplasm itself, you usually have reducing chemistry. And actually, if you even look at like the citric acid cycle, and if you look at glycolysis and things like that, it's actually, glycolysis is actually doesn't change the redox state that much. If you're reacting with oxygen, you're really going to be reacting in this special compartment. Even, if you have special oxidizing chemistry, you have a special organelle for that. That's what the peroxisomes are. So you seem to have a separate room for oxygen chemistry. And in fact, R.J.P. Williams, this is his exact words, he says, this was a major necessary strategy of eukaryotes because they had to reconcile a reductive cytoplasm with an oxidizing environment. So what they did is they made little oxidizing rooms. And that's a chemical way of looking at all this diverse chemistry, all this diverse biology under one chemical theory. So you can have the different compartments. Once you have them set up, they need to communicate. This is true for organelles and it's true for organs. And so if you look and you organize them as far as their chemotypes in increasing complexity, you can look at the different messenger chemicals that are required to communicate between the different structures that you have. If you set up an oxidizing room in a peroxisome, you have to somehow talk to it because you have that membrane there that's keeping all the chemistry safe, but you have to be able to tell it when to start and when to stop. And it turns out that these two appear to follow some kind of chemical rules that goes along with the general oxidation of the environment. So if you look at this, for instance, anaerobic species talk to each other with iron and magnesium, and they pass CAMP back and forth. Aerobic bacteria add on oxidized molecules, sulfate, nitrate, and things like that. Single-cell eukaryotes, when you start to have these oxidizing things, they start to use calcium to talk to each other. What's really interesting is they use calcium to communicate, right? What's one of the factors in the Cambrian explosion? You see a lot of calcium-containing fossils. And the idea is that calcium made the complex life possible, and it made communication within the complex life also possible. That's why calcium is such a big player in chapters 9 and 10. Not before that, but only at that point. If you move on to multicell eukaryotes, you start to get zinc and copper playing the roles that were on the last one. And when you start to have multi-cell eukaryotes with nerves and brains, you really start to have organs, a different kind of organ that uses a different kind of chemicals to communicate. Multi-cell eukaryotes are going to be talking to their liver on a slow time scale with zinc and copper. Multi-cell eukaryotes with brains and nerves start to use sodium, potassium, and chloride. And the reason why is one of the big things in chapter 10. Realize also that organic chemicals that are often using oxygen in some way, like an old molecule like decorated with oxygen, organic chemicals show up in this scheme between calcium and between the halides, between the sodium, potassium, and chloride. So remember that you have these organs. Organs are bigger than organelles. They're made of collections of cells, and they require specialized communication. If you're talking about hormones in biology class, realize that those are usually these small organic compounds that are usually an old amino acid that's been sort of messed up a little bit with oxygen. It's just been reacted to make a small molecule, and the whole point, that small molecule, the hormone, is a communication vessel. It allows your thyroid to tell your kidneys what to do. It allows the different organs to grow up at the appropriate time, because you don't need all these organs right when the organism is first born. Instead, they need to grow and develop. And that growing and developing is communicated by small organic molecules, copper and zinc. You see copper and zinc right here. So the adrenal glands, the pancreas, these are all parts of the body talking to each other, and you have to get everyone on the same page. The chemistry is used to get everyone on the same page. And so here's just another example. This was shown more artistically in the book by the illustration by Gayla Bent. But hormones will communicate between organs, and if you know your amino acids, all these hormones look familiar. Here's adrenaline on the top. Here's an animal hormone. Here's a plant hormone. These all look kind of the same. These all look like different amino acids, and in fact, tryptophan looks exactly like this hydroxytryptophan right here. And that's related over on this chart. You can see that's related to serotonin. And that has to do with the whole myth that if you eat a lot of turkey has a lot of tryptophan, you'll make a lot of serotonin and you'll feel sleepy. It will communicate the sleep thing. That's actually wrong. There's actually not enough serotonin. It doesn't get to your brain fast enough. But there's other things. It's at least plausible that that could happen, you know, because amino acids right here on the left make all these hormones that do all these different things on the right. Even the process of making those hormones is usually mediated by either iron or copper. And even in some cases, selenium, which is another late appearing element. So if you want to look at the organs like a chemist would look at the organs, you would say, okay, they specialize in different functions and they have different elements. So you need to deal with food consumption. As a multi-celled organism, you need to take in the environment and you need to be able to consume those plants, right? So you need digestive systems over here, stomach and things like that. Those tend to use zinc and calcium. Pancreas, remember that? That had zinc. If you need to have like internal minerals, that's what bone is. This is a weird way of looking at bone, but bone really is a geological kind of thing inside you. If you need to have controlled growth, you use hormones and glands to do that. If you need to move, you use calcium in muscles to be able to move. But if you need to control the movement, you need even faster control of that. So you need something faster than calcium to be able to tell the calcium when to go, be able to tell your muscle where and when to move. And so all that chemistry works out. That is by function, organs organized by function. Here's organs organized by element. So you can say that the iron is really important in the liver, in the spleen, and in the blood. And that's actually a reason. The liver does a lot of cool chemistry that depends on the iron. Copper and molybdenum are also in liver. This is why kids hate eating liver, but you know, people, I guess if you need nutrition, you know, the liver is a good thing to eat, you know, because it has a lot of these metals in it. Needless to say, there are other ways to get it, and thankfully we have other ways rather than eating livers, okay? But if you look up and down, if you look at the bottom, cadmium and selenium, strangely, are concentrated in the kidney, although this might be related to the kidney's excretion function. You have zinc being used in these communication organs, the prostate, the muscle, the liver, the kidney, and the pancreas. They all use zinc. And remember, zinc's a major player that comes on the stage in these advanced organisms. You see it being used for this new chemistry when animals develop. So why do we have that? Why do we have those fast elements in the brain and things like that? Well, it actually can come down to Chapter 6, Chapter 7 concepts. Because if you organize the elements by their K on and their K off, and by the way, the thing that you're familiar with now is K on divided by K off equals big K, right? And you can see that right here. R.J.P. Williams is a great chemist because he knows this just offhand, but now you know where he gets this from. And if you look at these numbers, the K-ons, the highest K-ons are sodium, potassium, and chloride, the same ones that are used in the brain. They have a very fast K-on, but they also have a fast K-off. And so they end up with a K that's about average, normal. So they're not good for holding on to stuff, but they're really good at moving fast. If you look right below that, you have calcium, which is fast on and slow off. It has a 10 to the 9th K on, but a 10 to the 3rd K off. And so its K is actually higher because of the way the ratio works out. That's one of the reasons why calcium is used in the muscles. In the muscles, you need a good equilibrium constant because the muscles actually need to physically move from one place to another. So it's good to have a very physically binding element. If you look down at zinc, zinc's actually similar to calcium, except in its on rate, but it's different in its off rate. It sticks on a lot longer. And so that's used for hormone communication. It's also used in a minor way in the brain, by the way. I went to a talk recently at the ACS conference where the very prominent chemist got up and said, if I was starting over right now, I would study zinc in the brain because I think that that's going to be the next big thing. And what's really interesting, if you follow Williams' argument and you see that zinc is like calcium, you can say that makes chemical sense. Zinc is like calcium, but it's used in a different way, and it's used for advanced functions. It makes sense that it would be used in the brain. And so then you have the really slow arrow that goes with the really slow on things. Magnesium is just too slow on to be much good. It has a K, big K, that's similar to the top row, but its K on is so different, it's like 4 orders of magnitude slower. So you can see if you're working a Chapter 7 problem with these kind of things, magnesium would be the rate limiting step if you had the on rates that you're looking at. I think I have one more arrow here. There it is. Look at copper. Copper is fast on, but its K off is even smaller than 10 to the minus 7th. And so its big K is huge, but it never falls off. And that's the same kind of thing we've been saying. This is reflected in both the equilibrium constant, big K, and the kinetic constant, little k. But this shows you copper, again, is special and unique in its role that it does these things in. This is all reflected. These were processed, and I got the data from R.J.P. Williams, same data, but done much more interestingly. This is a graph, and to give credit where it is, it's Mary Chaffee. She was Mary Anderson when she did that, and she got married. And so she's actually a TA right now, so we can say that the TA actually did published graphs in these things. But you see, this is the same thing. Big fast on, big fast off for the sodium, potassium, and chloride. And then you have the things that are stickier, which is shown by them being pointier in these things. And you have the slower and slower off. So these are definitely their own category of element, and they are all used in the brain. If you look up and down, the numbers are actually different. The K-ons and K-offs are different just because of the charge that comes from their place in the periodic table. The ionic radius, the ionization energy, you can talk about those things, but the main thing is the ion in water is water surrounding the ion, and the water can move on or off the ion. It turns out that water moves on and off sodium really fast. Water sticks a little more to magnesium, and that's one of the reasons why it's slower for it to go on, because it's got to let go of water before it grabs onto a protein. And if you look over here, the slowest, like beryllium, aluminum, things that are too slow for us even to use, they don't let go of water very much at all. And this appears to go with the differences in the on rates. Remember that copper has a very fast on rate too, and so it falls under the fast category here. It just has a very slow off rate. So when you put these together, you can see that the brain and nerve gets its own kind of chemistry. Very fast chemistry, so it can respond quickly to the environment. And if you look up and down at the different kinds of chemistry that you see that there's zinc and copper all through all the slow chemistries. But if you look up here at the top, you see lots of fast chemistry, no zinc, no copper. Zinc is used for something, but it's used for sort of a different function. These mostly rely on sodium, potassium, and calcium. And so that is how the chemistry of the organs works.